All cells rely upon a molecule called ATP for energy. ATP stands for adenosine triphosphate. The name of this molecule reflects its structure, which is essentially a nucleotide. The structure of an ATP molecule consists of the monosaccharide ribose, 3 phosphate groups, and a nitrogenous base called adenine. The chemical energy in an ATP molecule is largely stored in the covalent bonds that hold the 3 phosphate groups to the rest of the molecule. When one of these covalent bonds is broken, the chemical energy is released for the cell to use for other purposes. The most chemical energy is released by breaking the covalent bond holding the 3rd or last phosphate group to the ATP molecule. As is shown in this slide, this results in a molecule called ADP, or adenosine diphosphate, the liberated phosphate group, and also the released chemical energy. Cells produce their own supply of ATP. In a eukaryotic cell, the process of producing ATP is called cellular respiration. In eukaryotic cells, 2 molecules are required for cellular respiration to occur. They are the monosaccharide glucose and oxygen gas. The products of cellular respiration include the ATP that the cells need, carbon dioxide gas, and water. Cellular respiration consists of a long series of controlled chemical reactions. These chemical reactions can be divided into 3 main groups, or pathways. The first is called glycolysis. The second is called the citric acid cycle, but can also be called other names like the Krebs or TCA cycle. The last pathway is called oxidative phosphorylation. Glycolysis takes place in the cytosol of the eukaryotic cell outside of the mitochondria. The citric acid cycle and oxidative phosphorylation both take place within the mitochondria. Each of the 3 pathways has a specific function in the overall process of cellular respiration. The chemical reactions in glycolysis split glucose molecules in order to extract electrons from them. The citric acid cycle consists of chemical reactions that extract even more electrons. Oxidative phosphorylation is responsible for making the bulk of the ATP that's produced during cellular respiration. It does so utilizing all of the extracted electrons from glycolysis and the citric acid cycle. Now let's look at each of these 3 pathways in cellular respiration a little more closely. As previously stated, the chemical reactions in glycolysis split glucose molecules in order to extract electrons from them. These extracted electrons are picked up by a molecule that acts as an electron carrier. Its name is NADH. Other important products of glycolysis include pyruvate or pyruvic acid, a small amount of ATP, hydrogen ions or H+, and water molecules. Before the citric acid cycle can begin, pyruvate or pyruvic acid, one of the products of glycolysis, must be imported into the mitochondria. Once the pyruvate is in the mitochondria, it is converted by chemical reactions into another molecule called acetyl-coenzyme A, or acetyl-CoA for short. Each molecule of acetyl-CoA goes through the reactions that are part of the citric acid cycle in order to extract more electrons. These extracted electrons are picked up by electron carriers like NADH, as was also found in glycolysis, and a second carrier called FADH2. These 2 electron carriers, full of electrons, are the main products of the citric acid cycle. Other products of the cycle include a small amount of ATP, hydrogen ions, and carbon dioxide gas. This picture shows a model of a mitochondrion. Notice that it consists of 2 layers of plasma membrane, a smooth outer one, which is shown in brown, and a very folded inner one, which is shown in red. NADH and FADH2 from glycolysis and the citric acid cycle deliver their extracted electrons to the very folded inner membrane of the mitochondrion. They drop these electrons off to molecules that make up a chain called an electron transport chain that is embedded in this very folded inner membrane. The electron transport chain is the first part of the pathway called oxidative phosphorylation. The folded inner membrane of mitochondria is full of electron transport chains. Each electron transport chain consists of a series of molecules that are designed to receive and pass on electrons, kind of like people receiving and passing on water as part of a bucket brigade. The last molecule that is part of the electron transport chain is oxygen gas, which is the oxygen that you breathe in. The purpose of passing electrons from one molecule to the next through an electron transport chain is to power yet another molecule found in the same mitochondrial inner membrane. It's called ATP synthase. ATP synthase is responsible for producing the bulk of ATP that is generated in cellular respiration. It does so by adding a third phosphate group to molecules of ADP. The ATP that results can be exported out of the mitochondria for the cell to use. Approximately 30 to 32 ATP molecules are generated for each glucose that enters cellular respiration. 2 of these ATPs are generated by glycolysis, another 2 are generated by the citric acid cycle, and the remaining molecules are produced by oxidative phosphorylation and the activity of ATP synthase. Recall that oxygen gas is one of the molecules required for cellular respiration to occur. It is the last member of the electron transport chain that is part of oxidative phosphorylation. If glucose is available but oxygen gas is absent, neither the citric acid cycle nor oxidative phosphorylation will take place at all. Instead, the only part of cellular respiration that will take place is the pathway of glycolysis. The repeating of glycolysis over and over again in the absence of oxygen gas is fermentation. Fermentation is a way for a cell to keep making ATP in the absence of oxygen gas. However, it is a lot less efficient at producing ATP than if all of cellular respiration was completed. For every glucose, only 2 ATPs are produced as a result of fermentation, whereas 30 to 32 ATPs would be produced for that same glucose if all of cellular respiration was done. There are 2 major forms of fermentation. One type of fermentation is called lactic acid fermentation, which is what your muscle cells do during strenuous exercise. Yeast cells do another type of fermentation called alcohol fermentation. Both of these types of fermentation allow cells to keep making ATP when glucose is available but oxygen gas is absent by repeating glycolysis.